Hello everybody. I'm going to share the information about the prokaryotic cell. So let's start with the definition. Prokaryotic cells are the cells that do not have any true nucleus and membrane bound organelles. So what is exactly the prokaryotic cell? Prokaryotic cell are the single celled microorganism known to be the earliest on the earth. Prokaryotes includes bacteria and archaea. The photosynthesis prokaryotic includes cyanobacteria that performs photosynthesis. A prokaryotic cell consists of the single membrane and therefore all reaction occur within the cytoplasm. They are free living or parasites. The characteristics of the prokaryotic cells are different characteristic features. The characteristics of the prokaryotic cells are mentioned as they lack a nucleus membrane. The second one is mitochondria, Golgi bodies, chloroplast, and lysosomes are absent. The genetic material is present in the single chromosomes. The histone protein, the important constituent of eukaryotic chromosome, are lacking in the prokaryotic cells. The cell wall is made up of carbohydrates and amino acids. The plasma membrane acts as a mitochondrial membrane carrying respiratory enzymes. They divide asexually by a binary fission. The sexual mode of reproduction involves the conjugation in prokaryotic cell. The structure of the prokaryotic cell. The prokaryotic cell does not have a nuclear membrane. However, the genetic material is present in the region in the cytoplasm is known as a nucleoid. They may be spherical, rod shaped, or spiral. A prokaryotic cell structure is as follows. The first one includes the capsule. It is an outer protective covering found in the bacterial cell in addition to the cell wall. It helps in the moisture retention, protects the cell when engulfed, and help in the attachment of the cells to the nutrients and surfaces. The second one includes cell wall. It is the outermost layer of the cell which gives shape to the cell. The third one is cytoplasm. The cytoplasm is mainly composed of the enzymes, salts, cell organelles, and is a gel-like components. Cell membrane. This layer surrounds the cytoplasm and regulates the entry and exit of the substances in the cell. Pili. These are hair-like outgrowth that attach to the surface of other bacterial cells. The sixth one is a flagella. These are the long structures in the form of the whip that helps in the locomotion of the cell. The seventh one is the ribosomes. These are involved in the protein synthesis. The eighth one is plasmids. The plasmids are non-chromosomal DNA structures. These are not involved in the reproduction. The last one is nucleoid region. It is the region in the cytoplasm where the genetic material is present. A prokaryotic cell lacks certain organelles like mitochondria, endoplasmic reticulum, and Golgi bodies. So the components of the prokaryotic cells have four main components. The first one is the plasma membrane. It is on the outer protective covering of the phospholipid molecules which separates the cell from the surrounding environment. The second one is cytoplasm. It is a jelly-like substance present inside the cell. All the organelles are suspended in it. DNA. It is the genetic material of the cell. All the prokaryotic possesses a circular DNA. It directs what proteins and cell creates. It also regulates the action of the cell. And the last one and the fourth one is ribosomes. The protein synthesis occurs in the ribosomes. Some prokaryotic cells possesses cilia and flagella, which helps in the locomotion. The reproduction in the prokaryotes reproduces in the two ways. Asexually, which is by the binary fission, and the second one is sexually by conjugation. The first one is for binary fission. In the binary fission, the DNA of an organism replicates and the new copies attached to the cell membrane. The cell wall start increasing in the size and start moving inward. The cell wall is then formed between each DNA, dividing the cell into two daughter cells. And the second one is reconjugation or recombination. In this process, the genes from one bacteria are transferred to the genome of other bacteria. It takes place in a three-way conjugation, transformation, and transduction. The conjugation is a process in which genes are transferred between two bacteria through a protein tube structure called a pilus. The second one is transformation is the mode of sexual reproduction in which the DNA from surrounding is taken from the bacteria cell and incorporated in its DNA. And the last one is transduction. It is the process in which the genetic material is transferred into the bacterial cell with the help of the viruses. Bacteriophages are the virus that initiates the process. So, the example of the prokaryotic cell are bacterial cells, archaeal cells, etc. Thank you very much.